I would like to welcome you to the first distinguished lecture of the new semester. It is my pleasure to introduce to you Professor Giulio Cerullo from Politecnico di Milano. There are many people here who know Giulio very well, because if you've ever come across ultrafast spectroscopy, it's, I would claim, impossible to have not come across Giulio's work or Giulio's name. Giulio comes from Milan and did basically all his career in Milan. He started with electrical engineering, got his diploma, if I'm not mistaken, in 1988, and then did a PhD. He had a stay at UC Berkeley in the 90s, and then he moved up the ranks, starting from associate professor to full professor. He was just recently, again, a Miller professor at UC Berkeley, and won the EPS Quantum Optics Prize in 2023. Again, he's world famous for ultrafast spectroscopy, but he has also become interested in the use of ultrafast spectroscopy for imaging purposes, and that's what's going to mostly tell us today. And with that, I'd like to on the floor to you.
Yes, thank you, Vaid, for the kind invitation, for the nice introduction. It's really a pleasure to come here. And I realize that I have many friends in Erlangen, not only Vaid, but many people whom I'm very happy to see. So, as Vaid said, my group in Milano mainly focuses on ultra-short pulse generation, ultrafast spectroscopy. But in the recent years, we also moved in a new direction of nonlinear microscopy for biomedical application, in particular Coherent Raman microscopy. And so this is the topic of my talk. And I think there's also, I'm very happy to come here because we discussed with Vahid that maybe we have a chance to collaborate on this topic, really given also the very strong link of Erlangen with real hospitals and real biomedical applications. So basically, I will introduce you to the topic of coherent broadband coherent Raman microscopy, and then show you our work in this respect. So this is maybe a bit superfluous, at least for the group of Vaid, the motivation to go to level 3 microscopy. So of course, fluorescence microscopy is a wonderful technique for cell imaging. It enables to visualize morphological details with very high sensitivity. However, it always requires to introduce some exogenous markers. They can be dyes or quantum dot, or even you can do endogenous markers like fluorescent proteins. And this can sometimes perturb the system. However, it has high speed, high sensitivity, and down to the single molecule level, and you can get beautiful images. However, there are some problems. So the addition of fluorescent markers may be difficult in some cases. It can give strong perturbation to the investigated system, especially, for example, if you want to investigate a small molecule. Maybe if you put a fluorescent marker, it will be even bigger than the molecule itself, of which you want to study maybe the dynamics. Then you have a phototoxicity. You can generate reactive chemical species under illumination. So you really want to develop intrinsic level 3 imaging methods. And of course, the HiSCAT that's been invented by Vaid is an outstanding example of these methods. However, sometimes one would like to add also chemical sensitivity. So not get a contrast not only on the refractive index, but also differentiate specific chemical species. And there is in particular one application that we are targeting, which is a very big application, which is histopathology for tumor identification. So there are 3 billion biopsies that are acquired every year from patients. And actually the technique is surprisingly old. It's since 130 years, nothing has changed. And it's called H&E staining. So what you do is you take the tissue biopsy, which is basically colorless, and then you stain it with two dyes, hematoxylin and eosin, that's why you call it H&E, and then you look at it, you take the slide with the glass slide with the tissue, you put it under the microscope, and then the histopathologist looks at the slide with the microscope and makes the diagnosis. So, of course, this is a rather tedious and time-consuming task, both for the staining and for the diagnosis. And now the number of cases, unfortunately, is growing worldwide, also due to the aging population. The workload on pathologists is increasing, the number of pathologists is decreasing. So one looks to find a more efficient way to do it. And actually, a first revolution was so-called digital pathology. So digital pathology is in principle very simple. What you did, the first step is still the same. You still stain the biopsy with dyes, with H&E, but then instead of putting the slide under the microscope, you put it into a machine, which is the digital scanner, which essentially acquires a high-resolution picture of the tissue. So you don't have any new information with respect to the standard H&E, but now you have a digital file, and of course you can send it to a pathologist who is far away without needing to send a slide. You can store it, and then you can use AI. The AI, of course, can look at the image and can provide the first diagnosis. However, this digital pathology, which is still in Europe and in the US, not very widespread, it is more widespread in China, for example, but still it has some limitations. So essentially, you only look at the morphology. So you don't get any chemical information on your tissue, but you only look at the morphology, and based on the experience of the histopathologist, you make the diagnosis. So can you get more information out of your biopsy? And of course, getting the molecular composition would be a very powerful approach. And one technique that is very well known is spontaneous Raman scattering. So spontaneous Raman scattering essentially measures the inelastically scattered light, the Stokes scattered light from tissue and cells. And this is an endogenous marker. It doesn't require you to add anything to your tissue. And it is non-invasive and non-destructive. So you simply, in spontaneous Raman, you illuminate with a CW laser and you measure the Stokes spectrum. And you have the so-called fingerprint region, which corresponds to highly delocalized modes. And then you have a very intense region, which is the H stretching, which is in a limited wavelength range, where you have essentially the CH stretching bonds. And then you have what is called the silent region. So this is a very powerful technique, and it has been known since many years. This is now a typical Raman response of a cell with highlighting the fingerprint, the CH, and also a strong response of water. Now, it has been used by many groups to identify tumors. And actually, the idea is that you can make more or less the equivalence of an H&E image, where for every pixel you would acquire a Raman spectrum. And then based on this information, you can, of course, analyze and without any staining, reconstruct the H&E staining or go even beyond. And now for many kinds of tumors, this has been shown to have identified the tumor with very high sensitivity and specificity. So the sensitivity means that you identify all the disease cases. So everybody who is sick is correctly identified. But also specificity is important because you don't want to identify as sick somebody who is healthy, which is maybe not as bad as the contrary, but is still unpleasant that you get a wrong diagnosis in this. So you would like to identify all the sick people and only them. So I have 100% in this. And in principle, you can achieve very high values of both sensitivity and specificity. The problem is that why this technique is not used is because it's slow. So it takes you maybe 1 second to acquire a Raman spectrum, or a fraction of a second per pixel. Imagine you want to image a biopsy, maybe with thousands of pixels, so it will take you weeks, or it's simply impossible to do a high-resolution imaging with spontaneous Raman. So here comes into the play then the coherent Raman. So at the moment, let's say, if we make a chart where we put the imaging speed and the information content, with spontaneous Raman, you have a very high information content because you look at a lot of frequencies, so you get a lot of chemical information, but you are too slow for practical application. So now the other player is the Coherent Raman. So this cartoon tries to illustrate the principle of Coherent Raman. So what you're doing is spontaneous Raman. You come with a CW pump light and you interact with random thermal vibrations. So you are scattering off these thermal vibrations, and so that's why the signal is very low and the imaging is low. What you do with Coherent Raman, now you come with two pulses, the pump and the Stokes, and the difference frequency of these pulses is exactly matching the vibrational frequency that you want to identify. In this case, then now you make all the molecules to vibrate synchronously. So you create in the focal volume a vibrational coherence. And this increases the signal by 5-6 orders of magnitude. This means that instead of a second, you can image in microseconds. And so you can now really scan, as I will show you, a real biopsy in maybe minutes or tens of minutes in a realistic time. Of course, this comes with a price, and the price that now you don't have one color, but you need to have two colors, and these two colors need to be synchronized in time. So it is technically more difficult. Now, what are the two? There are two techniques of Coherent Raman. There are also others, but the most important are, the most well-known is Coherent Anti-Stokes Raman Scattering, or CARS, which was actually invented at Ford Motor Company in the 60s. And so what you do, now I'm showing you the simplest version where you have two narrowband pulses which are detuned in frequency by the vibration that you want to identify. So it's a K3 process, it's a third order nonlinear process. So you have one interaction with the pump, one with the Stokes, another interaction with the pump. All of these goes to virtual levels. So basically you are working in the infrared where there is no absorption. And then you generate your anti-Stokes light. So essentially you are generating a rather weak signal at the frequency, which is the anti-Stokes is the pump frequency plus the vibrational frequency. So is upshifted in frequency. So this is easy to detect because in principle, what you can do is you can put a filter, you can remove the pump and the Stokes, and you just detect your anti-Stokes. Now, what is the problem of CARS? So when in the early 2000s, people started to use CARS microscopy, there was a lot of hope. But however, people realized that there is, if you do it in this way, there is a big hurdle. Now, what I can do is I can rearrange these arrows in this way. I move the pump here. I move the Stokes up here. I move the anti-Stokes. So now you see, I am doing again a four-wave mixing, a K3 process, but now I'm not touching, I can even remove now the molecule, and I'm doing a non-resonant, this is a purely non-resonant process. This is a non-resonant four-wave mixing process, which is called the non-resonant background. And so anything in your tissue, in your cell, which has a non-resonant K3, which means everything that you have around, will give you this signal. So when you measure in CARS, you get the superposition of the resonant and the non-resonant signal. So it is very hard to tell which is which. And this is the problem that kind of, there are several ways that you can remove it, but it makes things more complicated. Now, the other process, is a stimulated Raman scattering. So you still start from the same two pulses, the pump and the Stokes, which are frequency detuned by omega. And now what you are doing is essentially you are doing what I basically have done all my life, which is a pump probe. So you are essentially, you are going to a virtual state and you are absorbing, let's say, the pump and you are emitting the Stokes. So you are amplifying your Stokes. This is so-called stimulated Raman gain. And you are depleting your pump. This is stimulated Raman loss. So you can choose to detect either of these signals, either of them works. Now, what are the advantages of this approach? You can demonstrate, it is easy to show, that now this is free from non-resonant signals. It only gives you a signal, at least in principle, when you may have some spurious effects, but it mostly gives you a signal only when there is a vibration. So it is free from this non-resonant background. And the other big advantage is that this is linear in the sample concentration. So it allows you to quantify the sample concentration. So it is the technique of choice. However, it also comes with a price. And the price is that this signal is typically, in practical cases, very small, on the order of 10 to the minus 4. So you need to fight with noise, with laser noise, to measure this very tiny variation in a short time. So you need to use some modulation transfer techniques. You need to modulate at high frequency and demodulate. So now these are the two basic techniques. And now, what is the basic layout of a single frequency CARS or SRS microscope? In principle, it is very simple. So you have a pump and a Stokes pulse, and they are detuned by the vibration that you want to measure. So at least one of them needs to be tunable in order that you can address the frequency that you want to measure. So first of all, you need to synchronize them with a delay line. You need them to combine them collinearly with a dichroic splitter. You have an acousto-optic modulator that modulates at high frequency, maybe a few megahertz. The pump switches on and off. Then you have a microscope objective that focuses into the sample, pump, and Stokes. And then at the output, you have a low-pass filter that only selects the Stokes. And then you have a lock-in amplifier, which is synchronized with the acousto-optic modulator and synchronously detected, for example, the stimulated Raman gain. And then you scan your sample and you acquire your image. Now, if you want to go from SRS to CARS, what do? You simply need to get rid of the lock-in amplifier. You get rid of the AOM. You change the low-pass filter with a high-pass filter, because now you want to detect these anti-Stokes. And you simply detect, let's say, your light. So here you don't even need to do any modulation. In principle, you can have two lines and you can detect both signals simultaneously after your sample. Yes, Farid?
Detector.
It will be a PMT, or it will be a more sensitive detector. It will not be the same for sure. Yes, a good point. But so in principle, you can decide. And now both CARS and SRS, especially SRS, I must say, which has been pioneered by Sunny Shi, have been used widely in many applications. So this is an example of label-free imaging of live cells, where you can mainly working in the CH stretching region, because it gives a much stronger signal. So you can detect DNA, proteins, and lipids by tuning at different frequencies. This is a measurement live of a cell division in cultured HeLa cells measured by SRS, where you observe the movement of the chromosomes by DNA contrast and the lipid contrast. You see it's here in green of the membrane. So you see it can be really fast. And then another big application is, of course, histopathology. This is one of the pioneering results by the Shi group, where they showed the label-free SRS imaging of a mouse brain tumor. So now you can really scan a tissue biopsy at a very high speed and get this very rich information. Now, yes?
Different frequencies?
In this case, you look essentially at two frequencies, one for proteins and one for lipids. Yes. Yeah, of course, you can... all this is done by tuning... measuring one frequency at a time, and then you can acquire images on multiple frequencies. Now, I just want to give you one example that was done by one work that was done by our PhD student Arianna Bresci, who is now at MIT, looking at monitoring cell senescence with coherent Raman scattering microscopy. So cell senescence is a problem that is encountered by human cancer cell when they undergo chemotherapy. So, of course, you would like to have therapy-induced cell death, which, of course, leads to cancer suppression. But there is another modality, which is what is called therapy-induced cell senescence, so that the cells become kind of dormant, senescent. So also the cancer is suppressed when the cells become senescent. But then the cells can come back, and then the cancer can relapse. So it's very interesting for a medical doctor to identify when a cell enters this senescence mode. And the question is, can we use coherent Raman microscopy to monitor the effectiveness of chemotherapy and identify the switch on of cell senescence? So actually, when we do this microscopy, it's actually a multimodal microscopy, because we can also we can not only do stimulated Raman scattering, but we can also do, for example, second harmonic generation because we have the short pulses, and we can do 2-photon excited fluorescence from endogenous fluorophores. So this is now what was found out is that if you take untreated cancer cells and compare them with chemotherapy-induced senescent cells, and there is a senescence-induced drug, which is the ferroxamine. So the senescence causes the accumulation of lipid droplets and the aggregation of the mitochondria. And actually, by doing a multimodal imaging, where we superimpose SRS, in this case at 2 frequencies, it is a narrowband SRS with 2-photon excited fluorescence to look at the mitochondria, you can really see quite clearly these hallmarks of senescence, which is lipid droplets accumulation and mitochondria aggregation. And actually, the medical doctors were very happy because they found from our images that they could identify senescence already 24 hours after treatment, while with conventional techniques, it would take them longer. So in this case, you can use this multimodal nonlinear microscopy to face a real biochemical problem. Now, going back to this plot, we say that spontaneous Raman is very rich in information. Yes, but
Could I ask how exactly you detect that in your, what's so specific?
Well, no, I mean, in this case, we look at the image of the cells. So we tune on the lipid droplets. In this case, you see the red channel are the lipid droplets, and you see that they are much more in the senescent case, and they become then after 7 days, they are really dominating. And then we look at the mitochondria. In the mitochondria, we detect them with 2-photon excited fluorescence. And then putting them together, we can extract, we can identify this difference, let's say, between the two kinds of cells.
You're not looking at it as shift the Raman.
No, in this case, not, because actually we are detecting Raman only at specific frequencies. Okay. Now, let me go back to this slide. So in spontaneous Raman, we have very rich information content, but very low imaging speed. In coherent Raman, we have a very high imaging speed, but if you measure at a single frequency, you have a relatively low information content. So you may want to tune it to get... So ideally, we would like to have the both worlds. So we would like to have a high imaging speed with coherent Raman, but also a high information content. So we would like to make broadband coherent Raman microscopy so that we acquire for every pixel of the image a full Raman spectrum as fast as possible, of course. And our dream when we started this work, and I will show you that maybe now we are approaching it at least, is to do what is called chemometric histopathology. So the idea is that we take a tissue slide. Here I'm showing you a standard H&E. And for every pixel with high spatial resolution, we acquire a Raman spectrum. And then this gives us a huge amount of data. It's a hyperspectral data cube because you get for every pixel a spectrum. And then you can use these not only to say tumor, no tumor, but also to classify the type of tumor, the stage of tumor. So to get as much information as possible that can help the diagnosis of the tumor. Now, so how to get a broadband coherent Raman spectrum? So there are these two possibilities, CARS and SRS. So I will start with broadband CARS microscopy. This was work led by my colleague, Professor Dario Polli, in the framework of this European project, Crimson, which included the group of Jürgen Popp and a few other groups in Europe. And the goal is really to develop this broadband coherent Raman microscopy. And we had a really exceptional PhD student who is now with Hervé Rignault, Federico Bernuccio, who did all of this work. So essentially, how can you do broadband CARS? There are two approaches. You can do either hyperspectral or what's called multiplex CARS. So in hyperspectral CARS, you have two narrowband pulses, and then you simply scan one of the two. And then for each frequency, the tuning, you acquire an anti-stock signal, and then you get your spectrum. But the more efficient approach is what is called multiplex CARS, where now you combine a narrowband pump with the broadband stalks. And then you are doing in this kind of process, and then you get simultaneously your anti-Stokes spectrum at all frequencies. Now, in this case, we need to combine two pulses, a narrowband and a broadband pulse. So how do we do it? We start from a laser, it's this laser, maybe I can show it here. It's a Coherent Monaco. I've seen it in Daniele's lab yesterday. It's a terbium laser, so it's a bit like the Pharos that I've seen in many labs here. It generates femtosecond pulses at 1035 nanometer with a very high repetition rate of 2 megahertz. And these pulses are maybe 250 femtosecond long. So what we do is, first of all, we take an etalon that narrows the bandwidth of the pump to about 10 wave numbers, so about 1 nanometer. So we have the pump is narrow and gives us the spectral resolution. And then we do a white light continuum generation in a YAG plate, because this laser is very powerful, so we can use simply bulk and nonlinear optics. We get a very broadband Stokes that goes from 1050 to 1500 nanometer. We need to compress it because this will be frequency chirped. Then we combine them with a delay line, and we focus the two pulses on the sample with microscope objective, and then we go to a spectrometer. The spectrometer is a CCD that can run, the shortest acquisition time is 0.8 millisecond. So this means that we can acquire approximately 1,000 pixels per second. Probably we could, if we get a faster spectrometer, we could run even faster, because our signal to noise would allow it. So what we do is essentially we scan our sample, and for every pixel we acquire a Raman spectrum. So we get our spectral data cube. We get hyperspectral data. We put them together. So for every pixel we get this raw spectrum. Of course, the spectrum is noisy. So first of all, we have trained a neural network to denoise the spectrum. And now this spectrum, you see, doesn't have the nice peaks of a Raman spectrum, because there is this non-resonant background, which is not chemically informative, which is overlapped. However, there are several algorithms that have been proposed that allow you to extract from this spectrum the real resonant spectrum, which is the imaginary part of the Chi3. And if you do this non-resonant background removal, then you get your spectrum, which is essentially like the spontaneous Raman spectrum, but now it's acquired in a millisecond, or in a fraction of a millisecond. So what you have gained is not information content, but speed. And this is true for any kind of coherent Raman. You don't learn anything different from spontaneous Raman, you just do it much faster. And okay, so then these are, for example, some sample. So this is now test on solvents. So you see the spectra that you measure are these ones in blue. So they are, it's not obvious to extract the features because of the non-resonant background. But if you do this algorithm, you can see what I'm showing you here is the imaginary part as a red, and the spontaneous Raman as a reference as gray. You see there is an excellent agreement in terms. Here we are limited to the fingerprint region, and these are all acquired with 0.8 millisecond acquisition time. And okay, we can do a concentration test and measure the sensitivity. So now, for example, we can take a sample of plastic beads with exposure time of 1 millisecond per pixel. Here I'm using two different plastics, PMMA and polystyrene, and the DMSO as a solvent. So we can essentially do this data processing. We extract the Raman spectrum. We identify the chemical composition, and then we can put them together, and we can make an image where for every pixel we identify the chemical composition. So here it's easy because you have only 3 compounds, so you can tell which is which very clearly from the spectra. And this image is acquired relatively fast. Now this is a more interesting sample. These are murine vertebrae. This is the H&E image. And now we can do the broadband CAS image. Again, we can identify, we can do a global analysis. We can identify characteristic spectra of marrow, muscle, and bone. and then we can do spectral denoising, extraction. And so you can see we can identify 3 characteristic Raman spectra of marrow, muscle, and bone. And then in this image, you can see really for every pixel, we can essentially assign one of these spectra. But of course, we can even use more depending also on our signal to noise. Now, with this, we are limited to the fingerprint. Now, if we want to access also the CH, the higher frequencies, we want to do what is called ultra broadband cars. And actually, what I've shown you is what is known in the literature as a 2-color mechanism. So essentially, I'm coming with a narrowband pump, a broadband stocks, and then I do this up and down, and then I generate my anti-stocks. So this is what is called an interpulse excitation, because to excite the vibrations, I need both the pump and the stocks. But there is another mechanism, which is known as the 3-color mechanism, where essentially I'm doing what is called an impulsive stimulated Raman scattering, that of course Daniele knows very well. So I'm coming with a short stocks, and the stocks goes up and down, and the stocks itself excites the vibrational frequencies, and then you can do you can come with a pump, and you can do then the anti-stocks. So actually, to excite, to generate this 3-color mechanism, you need a further compression of the stocks beam, because the stocks beam needs to be short enough to impulsively excite these vibrations. So it needs to be compressed to better than 20 femtoseconds. And indeed, if we now play a bit with our prism compressor, and you see we have a broadband stocks, now we can compress it to sub 20 femtosecond duration, and we measure it at the sample plane. And now we get these really ultra broadband cars, for example, on Tolwyn. This is now with an exposure time of 0.8 millisecond. And you can really see now the non-resonant background is this continuous red line. And now if you do the extraction, you get this beautiful Raman spectrum, which has to be compared to the spontaneous Raman. So now you see that you are essentially capturing all the frequencies of Tolwyn. So you probably have 10 or more peaks. And this is done again in 0.8 milliseconds. So much faster than you would get with a spontaneous Raman. And now, for example, we can do imaging of cancer cells, where we get both the fingerprint and the CH stretching region with 1 millisecond pixel dwell time. We can do imaging of tissues. This is non-alcoholic steatohepatitis, so it's a so-called fatty liver disease. So it's fatty liver that you get not from cirrhosis for alcohol, but simply from a poor diet. You get a lot of lipid accumulation, and indeed you can really see the lipid-rich end member, which is very apparent in this fatty liver mouse with respect to the control one. So we can also identify cancer on this is a comparison of an H&E stained slice. We can make a bright field image. You see the bright field image basically doesn't show you anything. And here with the CALS, we can get for every pixel a spectrum. And here we have, 3 millisecond pixel dwell time, which is of course good, but is still probably not enough at the moment if you really want to image a large tissue slide. So now we can go to the other approach, which is broadband SRS microscopy. This was again work led by Dario Polli with our postdoc Alejandro della Cadena. So now, how would you do the broadband SRS? Here we would combine a narrowband pump with a broadband stocks, and then we would essentially see the peaks of the stimulated Raman signal that are appearing on this broadband stocks spectrum. And so the problem is that these peaks are very small, maybe 10 to the minus 4, and here I greatly exaggerated them, but in reality they're really tiny peaks. So how do you detect them? So essentially what you're doing is you are combining your pump and your stocks. You modulate your pump with an acousto-optic modulator. They are synchronized on the sample. So after the sample, you select your stocks, and then you go to a dispersive element, for example, a grating. And now each frequency of the stocks is separated. So now we go to an array of photodiodes. So each photodiode measures a different frequency. So this is essentially a spectrometer. And each photodiode measures a frequency. Now you turn on and off the pump, and every time that the pump is on, you see these peaks coming. So how do you detect them? Essentially, you want to combine, to couple each of these photodiodes to a channel of a multi-channel lock-in amplifier. And then the multi-channel automatically does the subtraction between pump on and pump off, and you get your SRS spectrum. So this is the advantage that now this is background free. So there is not this non-resonant background. You get immediately the spectrum that corresponds to the resonant peaks that you want to detect. And if you can do it at high modulation frequency, you can really be very fast in imaging. So the problem is that this multi-channel lock-in amplifier doesn't really exist commercially. So we had to build one to do this experiment. And we were lucky that at Politecnico di Milano, we have really good electronic engineers. And so we had a collaboration where we designed a special lock-in for a multi-channel lock-in for doing broadband SRS. Now, the system that I'm showing you here is again a very complicated system. There's a femtosecond laser pumping an optical parametric oscillator, generating the pump and the stokes. So this is something good for our lab, but of course we cannot put it into an hospital. This is simply, it takes a whole optical table to do this experiment. This is, in this case, a coherent fidelity laser. So, and the OPO is on-built. So, I don't go into the technical detail. We had a really talented PhD student who spent his whole PhD building this lock-in amplifier, which is actually 32 channels in this case. And each channel demodulates at a few megahertz repetition rate the signal. So essentially, you can now get broadband SRS spectra of solvents in a very short time, in very good agreement with spontaneous Raman. You can do imaging of beads. This is a classical PMMA and polystyrene beads. You see that they have a really different SRS spectra, so you can clearly identify them. And here the pixel dwell time is on the order of, say, tens of microseconds. So it's really becoming faster. You can distinguish, in this case, we have four separate chemical species: olive oil, DMSO, PMMA, and polystyrene. We mix them, and every pixel of the image has a very clear spectrum that you can immediately recognize. So if you went like a single frequencies, you would have, in many cases, ambiguities. You couldn't really tell which is which, because it can be like a low concentration of one species, or so it's really important to have the spectrum for this. And you see that with 30, 40 channels in this range, you have enough. Then we did some application on, in this case, again, hepatic cells with an excess of fatty acids. So we looked at these EPA-RG cells which contain they were fed with two different acids, oleic acid and palmitic acid, which have different but not so much different Raman spectra. But again, by measuring the spectrum for every pixel and doing an analysis, we can really tell which acids enter the cells and into the membranes, and really look at the distribution. And we also looked at some images of tumor models. Okay, maybe in the interest of time, I'll go faster. Now, let me come to the most interesting part, in my opinion. Okay, what I've shown you is that the broadband SRS is now quite promising. It can be very fast, but it's too complicated. So here comes the last part of my talk, which is really the development of a real laser that can be put in a hospital. And this was done actually with the group of Andrea Ferrari in Cambridge, and we founded a company which we call Cambridge Raman Imaging, which is leading this European Innovation Council project called CHARM. And the goal is really to develop a microscope that can do this really fast histopathology. So what is the idea behind this work? So essentially, if you look at standard laser systems for cars, SRS, they are like the lasers that we have in our labs that I also play with all my life. They are bulky lasers. Maybe there's an optical parametric oscillator, so you cannot put them in the hand of a medical doctor. And they are also quite expensive. I mean, you need a specialized technician in optics to operate them. So the idea is to make a turnkey laser, and to these fiber lasers are really good. So the point is that you need to have always two colors, and you need to be able to synchronize two lasers. So the idea that initially we started to work on is that you can use graphene. So graphene is a peculiar material because it has these two direct cones that touch the direct point with the valence band fill and the conduction band empty. So you can do vertical transitions, you see, basically at any energy. So it is the only material that absorbs light throughout visible and infrared. So if you take a semiconductor, of course, it has a band gap, but graphene absorbs light at any frequency, at any color. And also it has a very fast, when it is illuminated, you bring the carriers in the conduction band, but the carriers thermalize very fast. And so the absorption saturates and recovers very fast. So it's what is called a saturable absorber. And it's a broadband saturable absorber because it works at many wavelengths simultaneously. So the idea that we had with Andrea Ferrari was to create a dual color fiber laser where you have an erbium fiber and an ytterbium fiber laser which share a common arm which contains the saturable absorber. So we started with graphene, but then we came up with some different solution, which is a bit confidential of the company. But in any case, the idea is that you have a common saturable absorber that somehow it mod locks, that it brings the laser into pulsed operation, but also synchronizes them. This is essential because you need the pulses to come synchronously. And here you see really a synchronization. You see the two pulses of the laser on an oscilloscope, you see they are synchronized. And you see the RF bit knot is really narrow, showing a very high level of synchronization. And all these now can be engineered into a very compact fiber laser solution. Then you have, you can then amplify them with fiber amplifiers. This is rather easy until you get to hundreds of milliwatts of power, which is what you need for the microscope. So this laser now is really compact. We developed it, and actually this is how it looks. We call it a STRAIL, a stimulated Raman laser. And it is really designed, at the moment it works in the CH stretching, so it covers the range from 2800 to 3100 wave number. And one pump, the pump is narrow, and then the Stokes is broadband. So we can simultaneously detect the Raman spectrum at over 300 wave number region in the CH stretching. And it is designed with the power is even probably, we need even to attenuate it a bit when we go to this tissue in order not to disturb it. It's more than enough for doing the microscopy. And so it's a completely engineered and turnkey system that doesn't require any operational intervention. And then the second key element that we designed is the detector. So we engineered, now we have 40 channels. So these are, you see, these are each of these balls are 4 independent, no, actually these are 8 independent lock-in amplifiers. So these are 40 lock-in amplifiers that are, actually we use only 38, but okay. And so now we can detect SRS spectra with a very short pixel dwell time. Here I'm showing you, for example, 3 solvents, ethanol, acetone, and methanol. You see the spontaneous Raman spectra as a dashed line, and the points are the SRS spectra. But these spectra are detected in 10 microseconds. So now we can combine the laser and the detector into a real microscope. So the microscope in itself is not particularly innovative. What is really innovative is the laser system. This is simply, there are some galvo scanners, and the detection system is in transmission. And this is actually a multimodal microscope. It can also do second harmonic generation to photon excited fluorescence. Now, let me give you some examples. So here we are looking, for example, at microplastic. We took 3 microplastics, PMMA, polyethylene, and polystyrene. And here we image at less than 10 microsecond pixel dwell time, meaning that we can acquire this image in a matter of seconds, essentially. And for every pixel, we get a spectrum. And here you see the spontaneous Raman spectrum of polyethylene, polyMMA, and polystyrene, and the SRS spectra as continuous lines. You see there's quite good agreement. And so you can clearly differentiate these microplastics. And these, looking at microplastics is, of course, a very big thing now. Now this again, we are looking at these senescent cells. We can basically repeat these experiments now with this very compact laser. But what is really interesting is looking at tissue. Now we can take a realistic biopsy. We can put it in our microscope. And you see, this is now already several millimeters squared. And we can get really rich images. Here I'm showing you just a few channels. I'm showing you proteins, lipids, and collagen and elastin by second harmonic. But you see the richness of information. And all these are acquired in a matter of minutes, let's say. Again, I'm showing you this NASH mouse liver with the control and the fatty liver. You see a lot of lipids, which very clearly show already by eye. You can see from the images the accumulation of lipids. Now, this is a real head and neck sample biopsy given us by the University Hospital of Jena. There is a group of head and neck surgeons who really would like to use this instrument. So this is the standard H&E staining. You see, you get these colors, and this is like a centimeter. Now it's a few, maybe 5 by 5 millimeters. So it's a really big biopsy. And this is now the same biopsy imaged with 10 microsecond pixel dwell time. And you see now I'm showing you just a few channels again, also some other nonlinear channels, but you see that now you can really acquire these images, and every pixel now corresponds to a spectrum. So now we are in the point where we really want to extract all the information, but you already see, if you take a zoom, what is how richer is the coherent Raman image. And now what the histopathologists typically do is they take this HLE stain, the slide, and they annotate it. Based on their experience, they say, okay, this is tumor, this is healthy. So already you see what we can do is we already by almost the color, we can tell a difference between the tumor and the healthy tissue. But what we can do is we can do what is called, of course, AI staining. So we can take, since the medical doctors are used to these colors, initially we don't want to change them. So we can essentially recreate from an unstained image exactly the same color. And of course we have much more, but at least we start on the common ground with them. We don't show them any Raman spectrum. So we can start from there. But then now we have the full digital data. So what we can try to do, and of course, we have not yet done it because we just developed this microscope. So now we need to generate a lot of data. We can train an AI to help in the, of course, the ground truth will be the diagnosis by the histopathologist. And this is now a first result, which I find very interesting. So this is the H&E slides. And the slides were given by the pathologist, and the pathologist annotated some areas of this slide. Then they gave us the same slide unstained, and we imaged with SRS. And now we trained an AI this is done in collaboration with Cambridge University. There is a very strong AI group there. And so they trained it to identify the critical, the tumor area. And so you see here, this area that was, this part of the tissue, which had already been annotated, there is a quite good agreement. But then it also looked at this area that had not been annotated by the pathologist. So then we went back to the pathologist and we asked them, is this correct? And they said yes. So this is the first result, but it's quite encouraging in the sense that we can really hopefully use in the future these to make, of course, we will never replace the histopathologist, but we can for sure help them to save a lot of time and make more accurate diagnosis. But of course, it will always be the histopathologist who will have to make the diagnosis. It cannot be done by the machine. So basically, looking at the state of the art of histopathology, there are these digital pathology systems that are, of course, easy to use because essentially they are making a photograph of the stained slide, but they are relatively low chemical information. Then you can have spontaneous Raman, which is rich of chemical information, but it is simply too slow. So our ambition is to go here. So to do a broadband SRS and have a lot of chemical information combined with ease of use and also speed, so that you can really use it in a clinical environment. And so our dream, and this is what also is the goal of this project, is really to do chemometric tumor diagnosis. So we take an unstained sample, we image it with our laser scanning SRS microscope. So we get now an image that contains for every pixel a spectrum, which contains the morphological information that is already in the H&E, plus the molecular fingerprint, which is not in the H&E. Then we get these data hypercubes, and then the automatic chemometric analysis by AI will, of course, differentiate between normal and diseased tissue, but then it will go beyond, and it will hopefully also identify the tumor type. So it will do molecular subtyping and also the tumor grade. And these are important informations, hopefully can be done in a short time, compatible with the real histopathology workflow. So, and we are already working towards this, and this is like a prototype. It's not yet, of course, we are, this is just the design. At the moment, we would like to build this real engineered microscope where essentially you just put a slide, you press a button, and you don't need to do anything. And then this will give you the diagnosis. Okay, so I hope it was not too long. So this brings me to my conclusions. So I showed you different approaches to broadband coherent Raman microscopy. So broadband CALS, broadband SRS with multi-channel lock-in, and finally broadband SRS with a fiber format laser that can go really into the clinical environment. And I think that now we can do high-speed chemometric images of cells and tissues with a compact turnkey SRS microscope. Okay, just one very last point. At the moment, we are only working the CH stretching, but we are already aiming to extend it also to the fingerprint, because this will, of course, this will require some laser development. Actually, we discussed with Nicholas today a possibility of a solution, but this will, of course, give even, but already like this, we can get a lot of information, but there will be even more. And with this, I thank you for your attention.
Thank you very much, Giulio, for this wonderful talk with lots of detail from physics, very interesting applications. I think everyone here was, I'm sure, satisfied with lots of interesting things that they learned. It's an excellent example of how instrumentation and high-end measurement can actually be applied for something that is completely beyond the usual realm of physics. So that's exactly what we would like to do in Erlangen.
If I can just add a very last sentence. In a way, I've been in the business of laser spectroscopy for more than 30 years now, so I'm slowly approaching the end of my career. And I would like, if I could do something really useful, I would be happy. I mean, if I mean that is one of my ideas, yes. But I don't know if this will be useful or not.
I mean, you've already colored your slides so that pathologists understand it. That's very useful. Questions from the audience? Yes. Do we have a Zoom session going? Yes. But we don't have the cube, right?
Hi, very impressive work. I was just wondering, since these are very short pulses, how much intensity you can put on the sample before damaging?
That's a good question. Of course, you can photo-induce damage if you pump too much. The point is that we are doing 10 microsecond pixel dwell time, meaning that we move very fast. So the damage, of course, depends on the power and on the radiation time. And we select a regime where we don't damage. So typically you can put, let's say, tens of milliwatts if you scan fast. If you stay maybe with 10 of milliwatts 1 second per pixel, you may already induce damage. But if you go like a few microseconds, and so there is a window where you can operate, and we have identified it. We also published a paper looking specifically at photo-induced damage by ultrashort pulses. And so it is an important point, but we try to stay in a safe regime, especially with live cells, because you don't want to you want to keep them alive and look at them. Yes.
But I think it would be fair to say probably that for pathology you don't care.
For pathology, maybe you don't care. Yes. But still, I think you can reach a regime where you don't damage.
Yes. Thank you for the talk. I was wondering how scalable this multi-channel lock-in amplifier is. So why do you settle on 40 pixels?
Okay, well, I think at the moment, well, first of all, it's a matter of cost and complication to build it. At the moment, we are in the CH stretching region, which is a window of 300 wave numbers. And so our spectral resolution is 10 wave numbers. And then I think with 40 channels, it's enough. And you see that we can reconstruct the spectra really well. But now we are targeting in the next generation, the fingerprint region, which is maybe 700 wave numbers, maybe from 1000 to 1700, something like this. And there we are targeting already 100 channels. So this is essentially every of these balls is 8 channels. So you simply need to add more. Of course, it will become more expensive. But in principle, you can consider that actually it's surprising. I mean, since it is maybe, you know, if you buy a commercial lock-in from Zurich Instruments, it will cost you maybe 20,000, 30,000 per channel. Of course, maybe that is most of, it can do more things that we don't need to do in but our lock-in is, we can make it at a significantly lower cost. So in principle, we can go to 100 channels, but potentially even more, I mean, yes. Then you have an FPGA that really gives you sense the spectrum directly, yes.
Other questions?
Thank you. I was wondering how small can the object be that you're imaging? So you showed microplastic below 1 micron, but how small?
Okay, that's a very good question.
You still get a signal, right?
Yes, that's a very good question, actually, because now we have identified microplastics as a very interesting field. So the microplastics, let's say, standard techniques now are people use either spontaneous Raman or FTIR to look at them. So with FTIR, you cannot see anything smaller than 20 micron. With spontaneous Raman, you can go to 1 micron, but of course, it will be very slow if you want to scan. Imagine that you filter water, you get, I don't know, a centimeter square, you get a lot of plastics, you want to see what they are, how big. And so with our technique, I think we can definitely go below 1 micron. We can probably, I mean, for sure we can go around maybe 300 nanometers. There was a paper actually that even ended up in the New York Times last year by Wei Min from Columbia, who really looked at nanoplastics in mineral water bottles. And he found that they were much more than people expected in plastic mineral water bottles. And so the New York Times took it as, you know, it's very dangerous now that we drink. And so then he could go to 100 nanometer, I think, but with a single frequency. What we can do is we can get the spectrum. So, but I think there is a big potential because the nanoplastics especially are very important. I mean, because they can enter the cells, and so people want to really understand where they are, and yes.
Yeah. Fantastic talk, Giulio. Just a semi-serious question, as I'm also looking for to do something useful with my career in 20 years' time.
Okay. You are much younger than me, so you have time.
Can you fantasize real use of higher order processes? Or do you see them as an academic exercise, as we are doing them now? Or do you see that we will be able to use like a higher order nonlinearities?
Like you mean higher harmonics?
Like not higher order nonlinearity, like a 5th order process or 7th order processes.
But for imaging or for in general for any application?
This is a very speculative question.
Okay. Well, I don't know, probably for imaging, I wouldn't say. For sure, I mean, maybe this XUV, intense XUV could be used for metrology. I think there is also a big, I mean, like a coherent XUV source could be interesting for imaging or even lithography, maybe. Yeah, this probably, I would say, is the closest I can think.
One last question.
Really impressive work. So one question about, it's sort of associated with the fingerprints that you mentioned. So how far you think this can go to differentiate different proteins, for example?
This is what we...
or lipids, for example.
Yeah.
You mentioned lipids,
yes.
proteins. So like how far can you...
I think the lipids, you can do them very well in the CH already. Of course, the more spectrum you have. And now I was discussing before lunch with Vaid exactly this problem, how much we can differentiate with the Raman different proteins. And that's an open question. I think it depends probably on the signal to noise of your measurement and maybe also maybe you can, again, using AI would be very important if you can maybe get some, again, ground truth and associate some Raman spectra of proteins. And then I think, well, this is I cannot, for sure, I mean, you can tell, you can differentiate, for example, in the CH, it's very easy to say lipids and proteins and maybe differentiate different kind of lipids, CH2, CH3. To tell maybe in the CH, I don't think you have the sensitivity to differentiate among one kind of protein and the other. But in the fingerprint, probably you have more of these. But this is actually maybe something that we could investigate together in the future, hopefully, yes.
All right. There's one question in the chat. There's a question, yeah. So here you are.
Okay, this is...
which kind of preparation?
Oh, okay. This is a very good question, actually. I didn't... let me see. Because actually, one thing is that actually these samples... okay, these samples are FFP, which means formalin fixed paraffin embedded. So when people take the biopsy, they first fix it with formalin, they embed it with paraffin, and then they cut a slice. And so typically when you do the Raman, you deparaffinize it, you take away the paraffin, but it's not a perfect operation. So, and you are still left with some paraffin, which is very disturbing if you do it at a single frequency. But what we found is that since the paraffin has a specific Raman spectrum, then we can work having the spectrum. And so this was the first, so we can work with both fresh frozen and the paraffin. Let's say there are 3 billion biopsies every year that are taken, and these are all FFP. So the fresh frozen is only done typically during is intraoperatory, during the surgery they do this. But the standard workflow will be FFP. And so I think this is important for us to show that we can work. And the cells, the other question, the cells, let's say, okay, we didn't, at the moment, we didn't yet develop an incubator to work with live cells, but it shouldn't be very difficult. We work with fixed cells. So, but I think this is a standard thing to develop, and it should not be a significant problem. But at the moment, we didn't yet do it to work really with live cells in time lapse. We took cells and then we measured them at different time points, but not continuously, because we would need to associate an incubator to our microscope.
Great. Then please join me in thanking Giulio for the wonderful talk and discussion.